Thank you very much. Thank you very much, Chad, for the super embarrassing introduction. Thank you very much to Anaïs. The two of you are doing a great job with the organization today. So yeah, I won't repeat my name, but I wanted to give you a bit of a flavor of how we think about using proximity-dependent biotinylation or BioID in my lab. And before we go anywhere fancy, I just want to remind you. Sorry, I don't know. Looks like I can move it, but not with the arrow. Okay. All right. So I know that everybody here is aware of that the cell, eukaryotic cell in particular, is highly organized. And for people that work on diseases, you will know that the deregulation of cellular organization underpins multiple diseases. And this is true for lysosomal storage diseases. This is true for cystic fibrosis. This is true for many different cancers. But what you may not have realized if you're not a cell biologist is that not all of the structures and not all of the organization in the cell are the same. So some of those structures are bound by membrane, lipid bilayers in general. That would include, for example, the mitochondria. But some of them are formed by what is now recognized as liquid-liquid phase separation or the formation of biomolecular condensate. And this includes structures such as the nucleus, the centrosome, stress granules, and Peabody's. Yet there are other structures that are formed by polymerization of repeat, for example, filament assembly in the context of different cytoskeletal structure. And yet, of course, there are protein complexes, and I think we're going to hear about that later today. What I want to bring home today, though, is that not all of the structures are created equal, and therefore they're not going to be as easily analyzed by different approaches, depending which structure you're interested in. And so, by and large, when people have been using mass spectrometry-based proteomic to define the composition and organization of different structures, they've been using a technique that require first to take a cell or tissue and lyse it, break it apart in ways that will solubilize whatever you want to look at without disrupting it. So, for example, if you're interested in protein complexes, you're going to do lysis under a certain set of conditions that are going to enable you to keep the protein complexes together and soluble in your lysate. And if you're looking at organelles, you're going to use different sets of protocols to do that. The next step is going to be to fractionate the cell lysate into its constituent. And again, the selection of the tool that you're going to be using is going to depend what you want to look at. So for example, if you're thinking about organelles, which is what we're going to think about today, you're most often going to be using centrifugation, density-based centrifugation, and you're going to make many, many fractions that will then be analyzed one by quantitative mass spectrometry. And what you're going to do then is you're going to use the profile of what has been identified in terms of protein across all of these different fractions and a guilt by association principle to uncover the composition of each of those structures. And so this is great in terms of approach. This also is fairly scalable. It enables you to do many different conditions, one by one, of course, providing that you have sufficient mass spectrometry time. And it is compatible with dynamic studies. So for example, when SARS-CoV-2 came about, a lot of people started using these kinds of approach to look at the perturbation of many organelles and structures by following virus infection. However, there's many drawbacks. Some of those drawbacks is that by the fact that you must lyse the cells and try to keep things that you are interested in profiling intact, you will get post-lysis artifact. Most often you have loss of association, right? So some of the organelles will get a little bit porous and they're going to be leaching their component, or a component of a protein complex that has a poor affinity is going to fall apart during the purification. But sometimes you get aberrant gain of interaction, gain of participation. You also have limitation in the resolution, so you can only separate a mitochondria so much, right? You are never going to be able to separate all of the different compartments of the very complex organelle that is a mitochondria. This approach also is notoriously bad for membraneless organelle that are formed by phase separation, because by definition, this is a concentration-dependent process. So if you end up diluting your lysate through this fractionation and through running through the sucrose gradient. It's really, really difficult to keep your membraneless organelle intact unless you trick the system and you throw some chemical cross-linker that are on themselves going to perturb your system. And lastly, if you're interested in the contact between organelles and structures, which is really important again for biology, well, by definition, you've broken down, right? Because you're separating them. So is there a better way? And many years ago, we got super excited. We actually read that paper in a bar with two colleagues from Toronto, Stéphane Anger and Brian Roth. As Chad knows, this is where science happens in Toronto. And we saw this paper by Kyle Roux and Brian Burke, which really changed the field upside down. And this was really a forward screening approach for proximity. And this was the first time that this approach actually was shown to work. So what it is a way to fuse a protein of interest, which in this meeting I can call a bait without having to explain what it is, to an enzyme, which is a biotin ligase. And the basic design is very simple. You pick that fusion, you express it in cells. You're expressing it at levels that are low enough to hope to not perturb the physiological localization of the protein of interest. And then what you do is you feed to the outside of the cells the substrate for the enzyme, which is biotin. So this gets uptaken by the cell, activated by the enzyme to reactive intermediate, which is biotinyl AMP, that then diffuses away from where the bait is located and can label covalently with biotin protein on their epsilon amine in a very close radius, which has been estimated to 10 nanometer, but do not believe that to be a ruler. And so we do the experiment for a long period of time so that we have sufficient accumulation. And then at the end of this, you can lyse the cells in as harsh condition as you want because you do not need to maintain interaction anymore. This is covalently labeled. So you lyse the cells in ways that will disrupt your protein complexes, your organelle, denature partially your protein, and then you fish using streptavidin affinity. This has really high affinity for biotin, all of those biotinylated protein, which then we digest directly on the beads with trypsin, which is the enzyme that most people use in mass spectrometry to identify peptide. And then we, of course, identify and quantify the peptides. Now you can imagine that you're expressing an active biotin ligase enzyme in the cell, and this is likely to cause a biotinylation of many non-specific things. So you need to have controls perform in parallel, and at the minimum, you should have a control that has your biotin ligase expressed at the same or higher level than as the most expressed base in your data set. Once you do that, you have a data set that you can then analyze, and we have developed, and I'll show you that on the next slide, we've developed tools with Alexei Nesvishky and Mike Tyers over the years called Saint that enable us to do that and put scores onto our interactors, proximity interactors. But the key point here that I want to bring home is that this enables us to separate the proximity detection. Things work together in the context of living cells. In fact, it works in living organisms as well, from the identification of what was in the proximity. And we've shown now, and many other people have shown, that this works in most organelles, whether they're membraneless and membrane bound. But in the case of membraneless organelle, this is really a winning strategy for us. And it repurposed our computational tool. However, this is not perfect. You're still tagging your bait, right? You're still putting a large protein tag onto your protein of interest, your bait, and this and the fact that you need to express it in the cell, even if you maintain the expression, may itself cause artifact. So can we flip that around? Can we say, can we acknowledge that we will never get the bait perfect, even if we do CRISPR screening, because we're tagging stuff? And can we use instead BioID to report on the cellular organization of the endogenous untagged proteins? And the answer will be yes, so in case I don't want to have too much suspense. So just to remind people, we've been developing, together with many other people, toolboxes. So we have a scoring tool, which is called Saint, which we co-developed with Alexei Nesvishky, and I've been continuing to optimize. That works better for spectral counting, which is what I'm going to talk about today. And essentially what it does is it captures the level of information that you have from your negative control in terms of how many peptides or how many spectra you detect for each of the test protein in the purification of the control versus the purification of the base, and essentially tries to separate the distribution and give you a fairly conservative estimate of when it thinks that the interaction are all right. All right, welcome back. All right, so I was just saying, like, this is actually a super helpful tool. So for you can download it on your web browser, and it allows you to call in gene name or whatever looks like a gene name or any of the other identifiers that you're interested in. And just do that from a double click or a control click, and that will essentially pull out information about a specific gene or gene product that you're interested in, and you can tailor that. So this is actually quite helpful if you have long lists of things and you don't want to go and navigate to like all of the different outside tools one by one. All right. So to go back to the BioID and membraneless organelle and how we're actually going to make sense of these data sets. So I want to acknowledge my colleagues in Toronto, Brian Rout and Laurence Pelletier, who did that to some extent before us. Essentially, they use BioID with multiple base to try to define the composition of the centrosome-cilia interface. And that was actually a really good first start. This was in 2015, but that really told us that we needed to do things a little bit differently from this pioneering work. And so what I'm going to highlight now has been published from many years ago, but it's really the foundation of how we think about BioID for looking at membraneless and actually for the cell organization. So this was a work of a current faculty member in Toronto, Jiyoung Yoon, who was then a postdoc in my lab, who had been interested in defining the composition of those two membraneless organelles, the stress granule and the Peabodase. And you don't need to know anything about their biology. All you need to know is their small membraneless structure that are formed by protein-protein and protein-RNA interaction. They're cytoplasmic. And they're in very close proximity to each other. And in fact, sometimes they fuse and sometimes they share components. So really defining their protein composition was really challenging. So you can do it by microscopy. Clearly, you see one of them is red, one of them is green. But to try to purify any of those structures and identify their component has been really challenging. So at the end of the study, we ended up looking at 140 Peabody and stress granule protein, as well as many proteins that we serve as beacons along the path of synthesis and processing of mRNA, because these have something to do with mRNA biology. And I just want to tell you, like, initially what we thought we would do. Initially, we thought we would take a couple of markers of Peabody, a couple of markers of stress granules, and look at their data and essentially find more of the like proteins. So it would look something like that. That's actually real data. It's just like I shortened the list to make it visible on the screen. So I took two stress granule markers on the top shown in green, two Peabody markers on the bottom shown in blue. And every time you see a map from me, bigger and darker means more. So that's spectral count mapping. So as you can see, the profiles are overlapping but different. And we have some proteins that are enriched with the stress granule base, some proteins that are enriched with the Peabody base. And lo and behold, the knowns are actually where they belong, right? The proteins that we know are localized to stress granule indeed are picked up by the stress granule base only, the ones Peabody, blah, blah, blah. And the ones that were known already to be located in both structures actually show up with both. But we have new stuff in there. And I'm just going to show you one. So what do we do with that? So we have this one new protein, KIA0430. At the time, it was not known. Then we ended up just cloning them and putting them into expression vector to do microscopy. So we validate by microscopy that the prediction is actually consistent. And we do that for everything. And I believe this is the last microscopy image I'm going to show you. We get about 80% validation of any of our prediction overall with microscopy. And this particular protein, after that, you can look at the function. This turns out to be a new component of the decapping enzyme, which is one of the activities of the Peabody. But again, this works fairly well when you have a few bases. But what happens if you have 140 bases and you try to look at the data together? Well, you generate a huge and ridiculous airball that means nothing, and that's the last of these airballs that you will see. So what are the alternatives to get spatial resolution here, which is what we're trying to do, right? We're trying to untangle the Peabody and the stress granule. There are many things that you can do in bioID experiment and that we do all the time for other projects, so that, such as analyzing specificity in the interaction profile, looking only at the most abundant interactor, scoring against compartment control, scoring against a mutant if you're looking at a disease gene. But for the purpose of the rest of the talk, I'm going to talk about what we use for looking at organelle composition, and it is to reconstruct interactions prey-wise. Why did we think that this would make sense? It came from this very simple observation that even for the two P-body baits in de Jong's dataset, which here are called DCP1 and PATEL1, if you look at the quantitative recovery of preys with each of them, while there are some proteins that are on the 45-degree angle, you will see that many proteins are not, right? They're falling more on one side or more on the other side. While this could be because we are not good at doing experiments, we thought that this was probably not the reason, because the stuff that fell closer to the pink bait, DCP1, were actually known interactor of the pink bait, while the stuff that fell the other way tended to contain interactors that were known for that green baits. And so if you think about what bioID does and this fact that it generates a cloud of activated biotin locally, and then the cloud of activated biotin diffuse, you can imagine that what we're doing is we're looking at the diffusion of this cloud of biotin away from the bait. So if you're this green bait, you will essentially generate those concentric rings of activated biotin from stronger in the middle to weaker, and the pink bait does that. So those proteins that fall on the diagonal, and this is just conceptual, we haven't normalized anything, would be expected to be equidistant to both baits, with stronger labeling happening when they're actually a bit closer, right? And then you could imagine that the proteins that are labeled better in one or the other ones would be, again, having a different distance relationship with those two baits. So that's very simple. And so we use that as the organizing principle for all of our data. So if you can imagine now that you're not interested necessarily in the bait, because I told you already that they're all lying, and you're interested only in the endogenous preys, right? So with just two baits like that, you can't get much more information than what I just told you. But if you have the potential to do many more baits, like 140 in this case, you will have a lot more information. So let's just go back to trying to figure out what happens to your endogenous baits, endogenous prey in this case. So imagine that you have those 4 balls, those are prey, endogenous, not tagged, and you're doing bioID with bait 1 and get a specific distance relationship of those prey to bait 1. You do it with bait 2, you get again those different relationship to bait 2, and you do it over and over again. Well, at the systems biology and computational biology, I don't have to explain to you that what you're generating now is a really good data matrix that you can use for correlation analysis. And because I'm not a computational person, I thought let's try Pearson correlation to look at this data set, and it works really nicely. I'll show you in the next one. But the key things here is now what we're doing is we're looking at the relationship between those endogenous not tagged preys, and that turns out to be amazingly robust to baits lying. And in fact, you could have baits that have no meaning and no endogenous localization normally being able to report on prey-prey relationship in a way that makes sense. So to get back to what it showed, now when we look at the Pearson correlation organization of this exact same data set that was a hairball, this is what it looked like. So all I want you to look at is on the left are the big structures, so the nucleus, the cytoplasm, mitochondria, and the ER membrane fractionated separately here. The small clusters tend to be protein complexes, but what we're looking at are those medium-sized clusters, which are, this is just co-enrichment there in this case, which are essentially our structures. So essentially the P-body is blown up here, and within the structure, we've profiled it with sufficient bait number that we have information about the organization of protein within this P-body structure. And this was to us very remarkable. You will notice that by the fact that the C0 complex, this deadenylase complex, cluster very, very closely with each other. The Argonaute protein that are involved in microRNA silencing cluster with their loader. protein. And on this map, for people that know the biology of or the interaction of any of those, proteins that form direct protein-protein interaction with each other tend to be also closely located. So this is actually quite useful. So we can take now these maps and put them back onto a cytoscape network. And now what you see is really the structure, right? You see the composition of those components, the communication between the different organelles. And this is true for cytoplasmic granules and body. We since expanded it. This nucleus in this case was undersampled. So we've hyper-sampled the nucleus in a recent story that we just put on bioRxiv last week for people that are interested. All right, so that's worked really well for membraneless RNA-containing structures. How about the rest of the cell? So what we did there is, instead of trying to go for higher density of markers, because it's a lot of work to make 140 cell line. Everything is in stable cell lines, right? 140 stable cell line and do the experiments all by hand. We wanted to do maybe a lower sampling, but to profile the entire cell. So what we did is we took markers that cell biologists are using for looking at different organelle by microscopy. And we put all of those markers inside cells, again, stable cells. We put 244 of those spread across the entire cell. Some of them we had already profiled. And we do a QC with immunofluorescence and mass spectrometry followed by Go enrichment for recall of base localization. So even though I told you that we are able to use bases that are not perfectly localized, for this particular purpose, we made sure that we were only using bases that made sense. And all in all, we validated 192 bases that report on about 40% of the expressed protein. And again, we're doing the same process, right? We're going to do a correlation view of the cells. We can see that, again, we have really nice architecture, both local and across all of the structures. And this is essentially what that looks like if we're looking at a Pearson correlation view. Now, instead of just having a zoom-in version of a few specific organelle, you have actually the representation of the entire cell with less resolution within organelle, but still enough resolution. So for example, at the beginning, I told you that by fractionation, you never get at the organization of the mitochondria. Not a problem here. We are able to pick up 4 different compartments of the mitochondria: the IMS, the outer membrane, the inner membrane, and the matrix. We also pick both the inside and the outside of the ER, and we see connection that show up between the ER membrane and the mito-outer membrane. And this is really cool, because I also told you at the beginning that contacts between organelle was really problematic for the fractionation-based experiment. So let's just zoom out on that particular thing. So we were able, looking at the data, to identify new mito-ER contact protein using just a cell map. And so how we did that is by looking at the localization on our map of known contact site protein for the ER and mitochondria. One of them is called INF1, it's involved in pinching. The other one's a GTPA, is called SAR1. And we noticed that there were other proteins that were located in the same areas, or even actually more in between those two structures. So we cloned them, we validated they localized there, and then we did both overexpression study and siRNA in the context of human fibroblasts for some of those components. And one of the hallmark of protein that locate at a contact site is they change the mitochondrial dynamic. They either make it longer, more fragmented, or they extend the network. And lo and behold, the proteins that we had identified are actually not only located at the contact site, but they have a functional role at the contact site. So this really highlights the power, predictive power of the approach in identifying new biology. We can also use a cell map to really look at identifying new, what we call sensor, to map the changes that occur at the surface of an organelle or in an organelle following perturbation. Perturbation can be anything: a chemical, a drug, a genetic aberration, and so on. And by sensor, we mean that this is not necessarily the protein that you're actually the most interested in, but what you want to do is to look at the recruitment or absence of recruitment of specific protein to your sensor. And if you want to read more about that, we published the first set of that in Science Now for the target of rapamycin signaling. Okay, so I told you that I'm not a bioinformatic person, and I use Pearson correlation for a long time. But then I was having beer with Quade Morris, and he told me like, yeah, but you can do better. And we suggest that you try to look at non-negative matrix factorization to essentially enable for looking at multiple locations for each of your prey protein. And it turned out that it worked great for us. And I'm just bringing that up here. This is essentially the NMF view of the cell map, which is now available on that website, Human Cell Map. And this is essentially what we're doing now for all of our studies, where the goal is to define the composition of specific organelles and also the propensity of any given prey protein to be part of organelle A versus organelle B. And so we're using that as a framework as we're expanding the number of studies that we're doing. But where we want to go from now knowing all of the concept that I've introduced here, and knowing that we're having this tool to analyze the data, we want to move a little bit further. The cell map we were very, very happy it got published where it got published. And for us, it was really the first step into thinking a little bit differently about biology. So the current map was only done in one cell type, the 293 cells. It's done under one condition, very static. It was analyzed on an older instrument. For people that know, this was on an Orbitrap Elite, which is like 3 generations ago, which means that the quality and quantity of the data is not great. We could do better today just by changing that. It was based on older, slower enzymes that took us at least 3 to 12 hours to get a strong enough labeling for these good baits. And the quantification was not optimal because it was based on just discounting approaches. We can do better. And why do we need to do better? Is we need to think about cell type specificity in the map, right? So how is the organization of cells different between different cell types? We need to look at dynamics. How do stress, stimulation, cell cycle, reprogramming, and differentiation alter the organization of all of those proteins inside the cells? And we also need to know whether we have essentially a predictive value from looking at patient cells, particularly in the case of mitochondrial disease that disrupt all of the mitochondria in unknown ways. And so if we do the same scale as we've done before, we need to have 200 baits, which in triplicase is 600 sample per bait. And we clearly don't have the capability to that, right? We're in Canada funded by the CIHR, so this would not work. So how do we take these goals and try to make it work? Well, first, we can optimize the number and the selection of the baits, right? So if we don't need 200 baits to get the same quality of the data, why would we need 200? Why would we profile 200 baits? We can optimize how we deliver the baits so that we don't have to create cell lines for everything, so that on the experimental list is a little bit better. We can select better enzyme now. It's in 2024. We have many variants to select enzymes that are more sensitive and enable us to get a faster labeling so that we can do the dynamics. And we can optimize the mass spectrometry. And I just will show you a few slides on that. So decreasing the number of baits has been accomplished in our lab by Vezal, who sits on the second row there, and had a poster this week. And essentially, since we know what we're looking for, which is essentially to maintain the structure that we get by NMF, we can essentially try to drop the number of baits that we need to maintain the structure, the resolution, the coverage, and so on. So Vezal showed, and he'd be happy to discuss to you, that using a genetic algorithm strategy, and he called this tool GeneBait, that he was able to drop the number of bait needed to keep the same coverage by threefold factors, which again enable us now to think about doing some of these experiments. For delivery of the getting rid of making stable cell line for everything, we published in 2018 the lentivirus delivery system that enabled us to do that. And this now enable a project that's led by Saya, right by Vezal, to essentially look at 5 different cell types and reconstitute the cell map without killing herself or me. But this needed to be optimized by the selection of the enzyme that we would use. There is currently 10 enzymes that can be used for proximity-dependent biotinylation. And so what we need to optimize for are many things, right? We want to use enzymes for all of our stated goals. So they need to be faster so that we can do the dynamic study. They need to have as low background as possible. they need to be able to respond to exogenous stimuli so that we can have temporal control, and they need to work across multiple compartments. So we did benchmarking across, we being Saya, Kozar, and other people in the lab, clone all of them, put benchmarking baits in there, and essentially we're trying to do this optimization, right, where we want to have highest signal for in the lowest amount of time and a really good response to the exogenous biotin. And so the basic idea is that we put all of the enzymes through their paces, and some of them fail because their signal was not strong enough. One of them completely failed at responding to exogenous biotin. Another one, which is by far the strongest enzyme, did not generate us a clean baseline. So for temporal control, where you need a clean baseline, this was not optimal. And then we had kind of the Goldilocks spot for two of the enzymes, which are Ultra-ID and Miniturbo, which were then used for specificity assessment with 15 of the 60 baits that we're going to be profiling. And long story short, in 10 out of 15 of the baits, it made no difference whether you're using Ultra-ID or Miniturbo. They're both good. They both give us good data. Oops, but, oh, I'm going the wrong way completely. But in 5 out of the case, including the one that I illustrate that, which is from Kozar, there is a major difference where Miniturbo failed to properly fold and failed to properly be trafficked, and then Ultra-ID is much better. So just on the enzyme selection, whenever you need absolute sensitivity, use Miniturbo, use Turbo ID. But if you are thinking about getting the enzyme that gives us kind of a better of both worlds, Ultra-ID seems to be a really, really good component. And we're going to be revisiting the background with each enzyme because they're all different. All right, I'm just going to go very quickly to say that the remaining bit of throughput acceleration that enabled this project has been more on the mass spec side. So for people that care, I'll be happy to talk to you. But we have been able to increase by 15-fold our throughput of sample acquisition by mass spectrometry, our sensitivity by many, many folds, and we're working on improving quantitation accuracy. And this now is enabling us to consider pilot project in reprogramming, which is a slow kinetic project, as well as fast kinetic project, for example, through growth factor signaling through the EGF pathway. I just want to tell you that we will need help from people like you that are looking, because the data now will be acquired in dynamic manner. We will be looking at multi-baits, so not just one data point, but multiple data points across multiple time points. And anybody interested in discussing that with us should, come to Toronto and have a beer, I guess. All right, so and last bit for people that are interested, we want to connect the intracellular cell map, which is what we've done so far, with some information extracellularly. And a student in the lab has also put a paper on bioRxiv that demonstrate the first use of this on our side for dynamic profiling. So I'm going to stop here. Thank you very much for your attention. Sorry about the little bit of that was really not my fault. I acknowledge people that have done the work in my lab, but the founders have really enabled this even in Canada. And people in the lab have been working together to make that better, and also with many collaborators in Toronto and beyond. And all of the websites are listed there for the tools. Thank you very much for your attention.
Thank you, Anne-Claude. We'll have a couple of minutes for questions here. I think we'll take a few because of the disruption. Questions, please step up to one of the mics, either this side or this side.
Sounds like it's on. All right. That was fantastic. So one of my questions is about resolution, spatial resolution and things like this. Some of these membrane-less organelles have subcompartments and things like that. So are you seeing some of these, and how well are you able to resolve them?
Yeah, so first, yes, in some cases, if we have enough density. And I highlighted that a little bit for the P-body, right? So within the P-body, we really see all of the complexes within that. For the larger one that you probably are talking about, things like a paraspeckle, and so we do to some extent, but we have not, for most of them, validated them by supra-resolution microscopy, which is what you would need to validate that what we see is actually, making sense. So for the knowns, it looks like it works. But for the new prediction, we've only validated one for a paraspeckle that is on the bioRxiv paper. But beyond that, I won't be able to comment. Thank you. Patrick?
Yeah, beautiful talk. Thank you. So for the identification of composition of these membraneless organelles or condensates, whatever you want to call them. So either if it's in different cell lines or in response to perturbation. So do you see changes that you cannot correlate with changes in the protein abundance themselves?
Yes. Short answer is yes. For a lot of the membraneless organelles, we do experiments in conditions where there is no detectable, at least, change in protein abundance because they're very short. Some of them could be for the most striking example is also on bioRxiv with a collaboration with Jason Moffat on a type of membraneless organelle called the twins body, where the addition of sorbitol for just a few minutes doesn't do anything to protein abundance, but completely changes the composition of, well, make them appear, the composition of those condensates. So yes, absolutely. In some cases, yes. And we can talk more about that if you want the details, but